Good morning, good afternoon. Thank you very much for joining our webinar dedicated to reshaping drug discovery with AI. My name is Anna Karawajczyk and I'm Director of AI and Computational Drug Discovery Unit in Selvita. I have over 20 years experience in supporting medicinal chemists with various computational tools during their efforts of transforming early hit discovery to a clinical candidate. Our webinar today has a following agenda. We will start introduction that will be dedicated to the AI in the drug discovery, followed by virtual screening within computer-aided drug discovery, and the abbreviation CAD you will hear very often today from me. The main subject of this webinar is TADAM, Target-Aware Drug Activity Model, the detailed description of how we have developed the model and what is the use of it later on exemplified in the case studies will be given by our experts in machine learning. TADAM will be also compared with other machine learning tools. And we will show you and talk about what other applications we have been using it in Selvita. At the end, we will have summary and we will open the discussion panel. Where do we go with artificial intelligence in drug discovery field? I would like to tell you at the beginning that all the questions that you have, you can ask in the chat, which is on the right bottom corner of your view, and we will take care of them at the end of the presentation. Computer-aided drug discovery has a long history, and generally speaking, its methods are complex and evolving with technological innovation on one side and the way how we see the nature on the other side. And artificial intelligence and machine learning, per definitions, are part of CAD, since those are simply the computer-based methods. Their application in the drug discovery coming to the target identification and validation, virtual screening and design, lead discovery and optimization, clinical trials, design and optimization is very much making their point in the field. And recently we are witnessing that the new models and new tools are entering the field of drug discovery, trying to reshape it or maybe even change the field as we know it so far. There are a huge expectation for that tool about faster timelines, of course, reduced cost, increased success rate, personalized medicines, and drug repurposing. All this we will talk today in our webinar, and we will present to you our tool. And this will be done by our experts in the drug discovery. I have a pleasure to present to you our experts in CAD. First is Dr. Jörg Wichard, who started introducing machine learning into drug discovery over 20 years ago, and now leading Selvita's AI ML team, and Dr. Michał Vieth, who has been assisting medicinal chemists over 25 years as modeler and the project leader, and now heading the group of structure and ligand-based designers in Selvita. Jörg, may I ask you to say a few words about your career and how do you see the growing significance of AI and ML in drug discovery?
Yes, thanks, Anya. So when I entered the computational drug discovery in Schering AG Berlin almost 20 years ago as a machine learner, we started with in silico tools for GPCR drug discovery. The databases were much smaller than today. The models were not so advanced and sophisticated, and let's say, coding a molecule as a graph neural network was a quite exotic endeavor. Nowadays, it's more or less the standard. And at that time, a lot of people thought the protein folding problem will never be solved because of the complexity and all these chaperones and what is there. A computer will never do it. Nowadays, we have, let's say, a Nobel Prize for AlphaFold, and it shows the significance of AI in the field. Yeah, so we were doing a lot of machine learning in computational drug discovery, and I paved my way then in computational toxicology, where QSAR is one of the things you want to do. And I always stayed in close contact with my machine learning roots. And 3 years ago, I entered Selvita, and I'm leading now the team of AI in drug discovery, and we see the growing importance, and I would say it is really reshaping the field, and later on we will see how we can screen these ultra-large databases, and only AI is enabling this.
Thank you very much, Jörg. And Michał, giving the voice to you, could you please say what was your career path prior Selvita?
I started at Lilly in '98 as a modeler, and then I started using actually machine learning the first year I joined Lilly, and it's been successful ever since. I'm more of a user of machine learning than developer. At Lilly, I also did chemoinformatics, bioinformatics, fragment discovery, medicinal chemistry, and then most recently I started using AI for structure-based design and coupling it with molecular dynamics and free energy perturbation, and also using it for de novo design of compounds.
Thank you, Michał. At this moment, there is also time to introduce Selvita to you. And Selvita is an European contract research organization established in 2007. We provide scientific support to various integrated drug discovery projects, from early hit finding till clinical candidate nomination. And with this wide introduction, I would like to invite you to have a closer look, together with our experts, to the aspect of hit finding, one of the aspects of the hit finding that we are using in the drug discovery, supporting the projects as a computational aided drug discovery group. I would like to give the voice to Michał and Jörg to start the main part of our webinar.
So when we think about virtual screening of libraries of compounds, whether real or virtual, there are two main avenues to go. One is using 2D methodologies, which is very quick, allows for screening of almost billions of compounds a day. And then the second approach is using a structure, target structure, for which we need to have a structure and a good hypothesis of binding site for multiple molecules. And for that, we use docking. And the throughput of docking is a lot smaller than for 2D methods. And we use a typical workflow where we initially dock the compounds, a subset of a collection, and then we select the hits and then process them, annotate them with properties, add me models many times. And then we go to a small number of compounds that we want to test experimentally. Well, it would have been great at the time that I was using it for a virtual screening to have a tool that can screen billions of compounds. And here it comes, TADAM, which allows you to use target receptor information as well as compound information.
Yes, so that is where the machine learning comes in. And as Michael mentioned, the docking is a computational expensive operation. And our idea was how can we screen these fast-growing virtual libraries a bit faster. And here I have the state-of-the-art on-demand chemical libraries in Q4 of 2024, so the most actual numbers. I think everyone who's a bit familiar with this field knows enamine, because they were the ones that started this, I would say, incredible track of billions of compounds that you can order, and 2 weeks later they are in your lab. Of course, you cannot order billions of compounds at once, but you can enter always a small subset, and that is what you want to test. So, and we have like e-molecules that gives you the building blocks for trillions of compounds. I mean, these are numbers that are really beyond what you can handle with docking. So our idea was, why not training our large machine learning model that can handle these large compound libraries. And we call this machine learning model TADAM, which stands for Target Aware Drug Activity Model. And the architecture of this model is described in the paper, and you can see the link on the lower corner, and it will be included in the email that reaches you hopefully tomorrow, where we will point also to this publication. You will find all the technical details there. But let me go shortly in the description. So as Michael mentioned, we have the 3D information of our ligand, that is the binding pocket, and we take everything that is in 10 angstrom sphere around the ligand, and this describes our pocket, and we coded bonds as a graph based on chemical bonds, and then we describe it also as a graph based on atom distances in 3D. This is the 3D information. And then we have to code also the ligand, and the ligand, we use the 2D structure, and we code it as a graph neural network. And here comes in that graph neural networks were developed in the machine learning community some, I would say, 20 years ago it started, 10 years ago they became popular. Nowadays we have whole libraries that are dealing with nothing else than this. And the training is well defined. You can train them quite well. You don't have to do it from scratch. And at the end, we want to predict the probability of a compound to be active. We trained this model architecture with this graph neural network with attention on a large data set that we collected from Campbell and PDB. It's about 900,000 ligand protein complexes that we put in the training of this network. And right now, we have a quite fast tool, and we can screen about 50 million compounds per hour. And this brings us really in the direction that we can go into an incredible amount of compounds that can be screened with this tool. So what we wanted to do is compare it to the state-of-the-art methods that are out on the market. There are two that are quite common, and you can find them in the GitHub repositories. That is DiffDoc and HyperPCM. Excuse me. And we compared them on, let's say, a data set that was not seen by any of these models. So therefore, we took the PDB structures that were released after 2022, and we got about 2,400 protein ligand complexes. We created also decoys that were non-active, and then we were running this test set through all three models. And after taking a look at the confusion matrix, we saw that if we scan them for recall specificity and the area under curve, and the TADAM is with recall quite much in front. Excuse me. This is something that you want to see, because recall is the key for high throughput screening, because you want to find the positive compounds, you want to find the actives. So with this in mind, we can see that our model is state of the art and can really compete with what is on the market. But that was not enough, because we wanted to have an experimental validation. And this is why we tested our model under, let's say, quite realistic conditions. We were targeting the JNK1 kinase. That is a target that we kept out of training. So this target was not known to our model, and we wanted to see how good we can extrapolate it. And we used our active docking pipeline that we had at that time, and we used our TADAM model in order to select 210 candidates from a virtual library. This library was created in the way that we took the building blocks from Woolport, which is an inventor of chemicals. These building blocks were then, in a one-step reaction based on Amit and Suzuki coupling, turned into a combinatorial library. And then we were selecting 210 candidates from our active docking and 210 candidates from TADAM and give them to our chemists. And our chemists were able to synthesize 45 from active docking and 55 from TADAM. And with these compounds that were then synthesized, we went into the biochemical assay. It was the JNK1, and that was a kind of HDS-like screening. That is what we do in order to find active compounds. So we wanted to see the realistic situation. And we identified several hits, and these hits then entered a detailed full dose response assay. At the end, we received 7 confirmed hits, and 5 of them were with an IC50 lower than 5 micromolar. These were the TADAM hits. And from our active docking pipeline, we had 1 hit. So this showed us at the end that our model works in practice. It was able to find active compounds against a target that it has not seen before. And that is really a good result. And we are, as you can imagine, quite proud to have it in our hands. Apart from using it as a virtual screening tool, we can use TADAM for different things. We can, for example, exploit fragment growing ideas in the pocket. We can use the protein representation that we have and look for similar pockets. We can use the compound representation that we get and use them for QSAR, or we can do reverse screening. So we can turn the screening around and say, okay, we have an active compound from a phenotypical screen, and now we want to know what is a possible target. And then we can screen this compound against all targets that we can create, in the sense that we can pack and ligate it in pocket and see are there unintended targets, are there potential targets, and this can give us an idea what it is. We call this reverse screening. So this is a TADAM in a nutshell, and what we can do with it. And to sum it up a little bit, we have an AI-assisted screening that can cope with the fast-growing virtual libraries. The TADAM model is able to screen 50 million compounds in less than an hour, which is what we have in our current hardware and what we can realize. We can use the representations for different things, like extended QSAR. We have shown that the model is able to really find hits, and they can be synthesized and they are active. That is really a validation that shows this is working. And at the end, I would say that AI-enhanced computational chemistry is the path forward if you want to deal with a, let's say, next generation virtual high throughput screening. And that is, in this case, AI-driven. And with this, I give the word back to you, Anja.
Thank you very much, Jörg, for the presentation and telling us a little bit more about TADAM. I think something which is also important to mention that the TADAM, it was the work which was funded by European funds given by our government, and all the work was within the grant that we have applied and we won some years ago. I would like to ask you a question, though, because you have mentioned on your slide about the representation learning of the molecules that was created while developing TADAM. Could you please tell me what other applications you're seeing in Selvita? So how do you utilize this representation learning, and why it's different from that's what is known in the market? Because when I think about small molecules, I think mainly about fingerprints or about smiles. What is new here in that representation learning, and how do we use it?
Yeah, thanks, Anya. That's a very good question, because when we were looking in this, it wasn't kind of a side effect. We realized that we have a nice representation of our molecules that is inside the network. So this is how the graph neural network sees a molecule. And in machine learning, this is called feature learning, which is an opposite thing than feature engineering. And in the fingerprints that you mentioned, we would call that this is feature engineering. You think about what are the functional groups and how can we put them together in a way that a machine learning algorithm can understand them. In this case, we take all the molecules that were used to train TADAM, and we got an internal representation that is something like a black box, but this black box is, let's say, related to biological activity. And we use this internal representation as a kind of new kind of molecular descriptors and put them in our hERG model, so the hERG channel, the hERG inhibition model, that is quite important off-target in drug discovery because you don't want to hit it usually. And our internal QSAR model for hERG inhibition is based on these descriptors, and they are outperforming the classical ones. So this is an example where the representation learning looks into a little bit of a deeper truth than what we know from the textbooks. And it's some people like it, some people don't really trust it, but it works. And that's how we use it, yes.
Thank you very much, Jörg. You have mentioned QSAR, and Michał, you stated that you are much more user of the AI tools. And then QSAR as such is already present in our computer-aided drug discovery ages. But do you see that this methodology that has been mentioned by Jörg is really changing or reshaping the shape of your daily work in terms of the QSAR use?
Well, it's been reshaping my work pretty much ever since I started doing computer-aided drug design. What changed recently is kind of marriage of AI with structure-based design. And I think that's where the future really is. And also using AI in creation of new molecules. So free energy perturbation as well. I mean, there are ways to create machine learning from free energy perturbation results, so you can apply it to virtual compounds. And I think my passion is really in de novo design, creating new chemical space using AI and free energy methods. And that, while not possible for all targets, once it works, it's just a great thing to have.
Thank you very much. You know, AI field is developing so quickly that it's really hard to follow the literature. And you both, Michał and Jörg, have many years of experience in the drug discovery. Our statement in the webinar title is that the AI is reshaping the field. And I hear from your comments that it's indeed the true, mainly because we are going with much higher speed. We are tapping into the almost countless data points. But I remember, like in 2019, I got a comment that we are trying to develop a hit, developing a lead for a certain target, and we were very busy with a good number of chemists and biologists for about 2 years. Our lead was already quite mature, but the comment was that just recently went out the paper that somebody using AI did it within 2 months. And as you could see, the expectations about having AI being able to come with a drug within months is somewhere in the air. Can you please comment on it, Jörg? Could you please be the first one answering this question?
Yeah, thanks. I will try to answer because it is, of course, the big expectation, as you said, in speeding up the process. But a lot of the long time that developing a drug takes is because of mandatory tests. So, as an example, if you have the 2 years carcinogenicity study, which is mandatory, it takes 2 years, and there's no way to speed it up. What you can do with AI tools is to build models that are so good that they most probably select the compounds that will pass these tests in a good way, and don't fall out in late stage failures, so that the adverse effects are, let's say, controllable and are almost not there. So this is a way where AI can help, but you cannot speed up a 2 years study because it takes 2 years.
Thank you, Jörg. And Michał, what do you think about it?
Well, what I think it's that every new technology, and AI is one of them, initially has a lot of hype. And then we go through a period of distrust, and then we start usually to make it practical. And I think we are right now at the practical stage.
I think that's a great statement. Thank you, Michał. We have also some questions from our public. I would encourage everybody who has some questions to ask it as well. And the question is as follows, that TADAM was, of course, trained on the PDB database. So that means it has seen a lot of complexes of protein targets and small ligands. And how does it would perform on the targets that are not really presented or represented or well represented in the our public domain? What about, for example, allosteric pockets, some cryptic pockets? Would it still work fine, or do you see some constraints of this model?
Oh, that is a very good question. So the cryptic pockets, I think they are hard to find. We have to trust the computational chemists that find these pockets and can put a ligand inside, because what we need for TADAM is this 10 angstrom area around the ligand in the pocket. So I think we cannot find with this a cryptic pocket, but as soon as you identify something that looks like a pocket, or you have an idea that this could be a one, then we can try our best to run it. And we have seen that we were quite successful on finding hits for a target that was not in the training set. So we have good hopes that we can do that again. But of course, the more exotic the binding pockets look like, the more challenging will be the endeavor.
Thank you, Jörg. And I think I have another comment to that question, that we do not try to predict how the new compounds are binding to the pocket. It's really quite challenging. We have made this quite challenging. We had trained and we have combined together two words, 3D word of the pocket description and 2D description of the ligands. And then what we are predicting is the probability of the compound being active for a given pocket, but not how this compound would bind. So that's why maybe we are a little bit less biased into that set of ligand or only visible ligand protein complexes. So the question has a second sub-question asking like if we have any other recommendation in the case that there is some allosteric modulation needed. And I think here we can ask Michał, because he has a very good track record in molecular dynamics, for example.
So allosteric packets are always a challenge. I think describing a packet or finding out what the packet is allows tools like TADAM to utilize existing data. I think for any allosteric packet, virtual screening is low probability. However, it may work. I'm a pretty big skeptic in general, but I think what I've seen here, the TADAM tool has ability to do it.
Yeah, I think with the allosterics, it's from my working with allosteric pockets. So it's one thing to find a pocket which is distinct from the one which is used as an enzymatic non-binding pocket, but the other thing is how to propagate the binding into the real action, which is distant from the place where you bind. This can, if you are not able to catch in nature and observe it well, it's not so easy to find this kind of pockets and really utilize them in the project. But yeah, you are right.
I think just to add to that, because you mentioned molecular dynamics, it is possible to generate all possible conformations of a protein, but you need a very long simulations. And there are techniques like Markov state models, which we stitch together shorter simulation into a long one and allow full conformation analysis. But that's, again, very long simulation to understand all of conformational states and then find the buckets.
Yeah, I think also there is people who are trying to use some AlphaFold, like state-of-the-art different folding of the protein to try to figure out if there is some possibility. So that's still AI use, and in that case, reshaping our field. I have a question. So can you imagine that 50 million of compounds in 1 hour, it's still not enough for our audience. And the question is if we would like to scale it even up, maybe using some cloud solution.
So just a short comment, 50 million an hour, that's 2.6 billion a day, billion in American meaning. That's quite big. And I guess if you distribute it on the cloud, you can multiply it by the number of the processors, I guess.
Yeah, that's absolutely true. Right now we do it on our own high performance cluster. And that's why we are saying right now we are doing 50 million per hour. And if we parallelize it, and it can easily be parallelized, we can do more. But I think then the next generation of virtual libraries will maybe be a gazillion or whatever comes next. So there will be the race is on, right? And there was this paper from the Raymond Group that say there is more drug-like molecules than you have atoms in the universe. So there is no end with this perspective, 10 to the power of 60, or what was the number? And they are all somehow possible. There are no limits. That's the short version of it.
Yeah, and still the hardware needs to also keep up with the tools that we are building, but I think they will. People and not artificial intelligence is also very smart in going forward and pushing the use of artificial intelligence. Thank you for the answers, and thank you all the people who has joined the webinar for asking the questions. I still have a few on here, and the next one is TADAM is of course dedicated to the small molecules. It was trained for the protein and the small molecules, but one of our participants is asking if it's possible to use AI for non-small molecules therapies like protein, monoclonal antibodies, or immunotherapies.
Yes, let me answer this. Let's say the language of proteins are the amino acids. And what we see right now is that people using these large language models, and they specify it on the language of proteins. And so they feed it with the amino acid code and want to get something out of this that looks very interesting. On the other hand, if you have a hammer, then every problem becomes a nail, right? So that was how we started to use the Smiles to Smiles autoencoder. And in the beginning, we looked at it and said, well, will this really work? And at the end, it worked. It started to create the first learned features. So interesting. I think there's something on the way, but it's hard to predict when, let's say, the first language model that can really understand and speak the language of proteins is there and is doing something that we haven't seen before. That is my short answer.
Thank you very much. Michał, would you like to add something to?
I think you covered it really well. It sounds potentially possible, but challenging. The way it is right now, I think will have to be completely redesigned for the, as Jörg said, for the protein language of proteins or descriptors possible.
Yeah. When we talk about different therapies, there is also a tool that you have your publication on already from your early path or career, which was dedicated to the peptides.
Yeah, we did AI-driven peptide design with the Charité some really years ago, and it worked at that time. And well, there's a lot of say research currently going on, and we are also participating in this. But yeah, the field is really moving, and it's moving, as you said, it's so fast that we have sometimes it's hard to read all the papers and to be involved in these kind of things. But there are models that are, let's say, dealing with peptides, and we are also playing around with that. Maybe this is what I can share. Yeah.
Yeah, thank you. Thank you to the audience also for the appreciation of the webinar that we are getting in the chat. There is another question. Probably you have seen and read the paper that was recently published about contrasting the well-established docking protocols coming from Glide, Autodoc Vina, or Smina to the DiffDock paper from a few years ago, showing that the comparison maybe was not very much fair to those old style CAD programs, and showing that still there are some limitation of the machine learning models. for establishing ligand docking predictions. And the question is exactly about it, but not towards the DiffDock or the GLIDE, rather about TADAM. So where is TADAM in that competition? Can we compare it somehow with any other methodologies that are available for the scientist in the public or non-public domain?
Yeah, I mean, the comparison that we did was with DiffDock as one of the two. Of course, there are many out, and maybe there are some that are better. We don't claim that we have, let's say, the philosopher's stone, and that's it. We are also thinking about retraining it on the next release of Campbell. We want to introduce some new learning features. We have some ideas how to extend it, so there is, say, a lot of space to go forward. And let's say we don't see it as a kind of thing that is now better than docking. That is not what we want to, let's say, this story is not what we want to tell here. It is one way to deal with this huge amount of molecules in a specific way. And of course, if you have a skilled computational chemist and a good docking program with all the long time experience and a very good X-ray structure, then of course you can do much different things and be more precise and specific. But you cannot do it with 50 million compounds per hour, right? And that is the thing. So we, I think the AI and the machine learning is helping in all stages of drug discovery a little bit and moving it forward. And sometimes you have these breakthroughs like AlphaFold was solving the protein folding problem. But you don't have that every day. So I see it more that we do what we can to play this game of drug discovery, and it is finding the needle in the haystack, and that AI is one of the many tools we have to do this job.
Thank you very much, Jörg. And I think something which we also should keep in mind that TADAM is predicting the probability of being of the compound being active.
Yes.
And it's really not predicting the conformation of the ligand inside the pocket. And the conformation of the ligand, it's still a problem to be solved. And having the conformation of the ligand, which is fitting to a protein that will be giving the action of interest, is completely different story. So that's why, as a computational chemist, we are taking the nicely tuned compounds coming from TADAM, and we are still doing a normal, well-established docking and molecular dynamics in order to figure out which compounds are the best in the selection to give to our medicinal chemist. As Michał has mentioned, there is another application of it with using free energy perturbation, which is even closer to the real binding affinity that is measured. So the field is going to the direction, but still there is some homework to do. We are slowly going to the end of our time for this webinar. So with this sentence, I would like to thank you very much for the participating in our webinar. Thank you, Michał. Thank you, Jörg, for taking part into it and sharing your observations and comments about how the AI is reshaping the world of drug discovery. Thank you very much for your attention, and we wish you a very good day. Goodbye. Thank you.